So that is actually part of what the Living Biofoundry is about. We are a robotic automated facility, basically, I mean, at the basic level, trying to get people from spending their time actually pipetting after they've had this great idea to actually being able to have robots that can implement that pipetting, but also bigger to be able to scale it up so that we can begin to think about biology in a 100 experiment at a time way to solve the biological problems rather than asking, did our assay work this week? Right? That's what we would try to do. And again, it takes longer to get a robust assay working. But when you have more and more and more of your component automated, you can start doing your science faster. So the real purpose of our facility is to actually accelerate the rate of scientific advance, not just have you not do as much pipetting. That makes sense. Both are good. So with that, I also, that's a Living Biofoundry. I should step back for a second. I'm also representing the rest of BioPACIFIC, where we're housed. So we are the production side on the Living Biofoundry to make monomers that actually have a further life that then go into polymers that are made by our collaborators at UC Santa Barbara. And so part of the schema that we operate under for the NSF is to basically try to create materials that would not be accessible if you did not start with things that could only be made in a cell, and then move them to a material that can only turn from a goop in a cell, some liquid you extract, right, into an actual hardened 3D material when you send it to polymer chemists like up at Santa Barbara. So we're going to talk about that nexus across all 5 of these facilities, starting with the automation design principles that I talked about, meaning we're trying to get higher throughput, more cycles, more shots on goal, and we're trying to take advantage of the idea that we can practice the science fast enough to also design the experiments to generate inference, inform, and initiate a new cycle that's even more productive than the last time we did it. The difference is when you do it the second time, the robots are still ready and waiting. The first turn of the cycle is much harder. So with that, this is the Living Biofoundry. Let me go ahead and, this is actually our facility upstairs. It's a Thermo Fisher lab automation system with a whole bunch of extra bells and whistles we've customized and added. If I start this video over here, this is what it looks like, taking a plate out of an incubator, moving it around. And while this plays, I'll tell you what we really are all about is, like we said, the DBTL cycles, what I've been talking about, is design, build, test, and learn, right? And I would say one of the most important things we are trying to work with our users is trying to get away from that linear model of research, where we set out to accomplish this goal, we do these experiments, we get our conclusion, we publish a paper, and then it stops. Oh, thanks. We want to return that to basically say, okay, we finished. Now let's input what we learned from that last cycle, repeat. And so we want your project, we want you to have the one extra thought. What are you going to do if you get 500 answers or 1,000 answers or whatever it is we're capable of delivering to you that then allows us to start the next stage of the experiment and advance and accelerate the development of materials, as opposed to fixating on, like I said, did you get 500 measurements, get some good data sets, and get a publication, right? And publications are great. You get them at each stage, right? You can publish at each turn of the cycle. That's fine. But acceleration is a different concept. So with that, we also have all these standard processes like PCR, automated liquid handling, plate readers, the standard stuff every biology lab already has and knows, but we're trying to integrate it into this more automated framework. So with that, I'd also adding on to what Adam said, that's the commercial stuff. It's all operating, come down, we can talk about it. And what I would say is, as you all are probably familiar with in the biology side, a standard set of equipment can accomplish an amazingly wide amount of goals in the biology space, right? This same robotics equipment is used for high throughput drug screening. It is used for COVID testing. It is also used for biomanufacturing, right? So we have incredible capability. The key is, can we thread everything you want through an automated platform so your entire assay becomes automated? To wit, one of the gaps we identified was that we had an electroporator we bought, which is the best in class, twin wave, works on yeast bacteria and mammalian cells, but was not fully automated, right? And the fully automated version that only works on stem cells is $150,000. So what we did is we actually built our own robot to press the button and a robot to raise and lower the actuator with an incoming freshman, by the way, in bioengineering, as well as a fourth-year physics student. We're also coding all the electronics. And that's our control electronics. I didn't show you the gory code, but we're also writing code with REST API. So it's actually going to be run out of our Thermo Fisher original suite, just like all the other professional devices in our line. So that's the idea. And I would just say to all of you who are interested in coming to do projects with us, we will take whatever idea you want to feed it into this infrastructure and maximize the impact of it. And if there's some specific gap that's just killing this amazing thing from happening, we'll try to build through that gap as well. I think that's what we mean when we say we're not just a place where you can come and do some push button stuff. So with that in mind, let me tell you one thing we're actually doing on the subject matter. And this is, for example, we do a lot of biopathway work. We make lots of small molecules. We are by no means limited to proteins, right? But as a protein example, This is a great example of what like a biofiber or a biomaterial of the future might look like. These are called SAM synthetic fibers. Also, we are expanding all the time our remote capabilities and our remote access. This is a collaboration we have with the University of Washington, St. Louis. These researchers are actually remotely operating our tools as well as collaborating and sending resources back and forth to accomplish this goal in our facility. And believe me, it'd be a lot easier if they could just drive over, like some of you that are local. But what they're doing is they're combining three super performers in the biomaterial world into one fiber that can outcompete cotton and nylon in some of the different categories, right? And that's because they're combining a silk fibroin, we've all heard of that. They're combining an amyloid plaque from Alzheimer's, which is very, very tough, with also muscle foot protein, right? The stuff that gets stuck on boats, right? Those are three huge performers. Put them all together. And then that's just the starting point. Because then imagine you're going to take that as your starting point and begin to engineer it and advance it through multiple cycles. Your later won't be any one of these three exact proteins. It will be the optimized phase space where you figure out how to get something softer and more flexible or something firmer, etc. Beat cotton and nylon in whatever your application is. That's the kind of idea for a higher tech framework for biomaterials development. So let me move on from that to where we talk about the automated synthesis. So this is where we get to automated synthesis in chemistry, right? We have at UC Santa Barbara a ChemSpeed, which is basically an automated suite that does all of the things you'd find on a bench in a synthetic chemistry lab, right? So analogous to what we were talking about with robots, but we're not going to see them today, so we're not going to go into as much detail. But it also is a glove box condition. You can have low oxygen environment. You can do things that we'll never be able to do. They also have a flow chemistry reactor, and they also have a peptide synthesizer, right? So you can imagine the flexibility of what you can actually produce, and where you can make new monomers, new peptides, for example, that can go into a new monomer for a peptide synthesizer that then goes in to make a new non-natural amino acid into an actual structure or into a material, right? There you go. So there's a chain from biology straight into that. We also have additive manufacturing, and here you can see we actually have this, one of the first collaborations with carbon to actually use the CLIP process, which actually generates things at the liquid-air interface, generates polymerization at the liquid-air interface, and it's 200 times faster potentially for making materials. We also have control. If you see those two dyes, that's Psi3 and Psi4, but what that is, think YFP, right? Your favorite protein. Right? They have a printer that can make sure your stuff ends up here and your other stuff ends up here, and it's structured into a polymer that can have the properties you want, hard, soft, flexible, what have you, right? So really amazing things. This is, again, what do you do when you take something amazing and make it in biology? You got to throw it into something that's structural and useful. So we have both shape as well as chemistry working in terms of the collaboration. High throughput characterization, as you guys have already heard a little bit, is another thing. What do you, again, design, build, test, right? Just like electron, great examples Matt gave of how important electron microscopes are for actually characterizing things. We similarly have an unbelievable best-in-class X-ray wax and sax line that, if you can see here on the bottom, is actually competitive with data you get from a synchrotron. It's not better than a synchrotron, but you don't have months of delay to set up. They run it fast and quickly and easily. They developed a higher brilliance source than any other labs, and that was the basis of creating a custom tool that is now widely available and under very high usage, but also very, very easy to use. You can literally mail them samples, get your data back, or go show up, get trained, and use it for yourself. So it's really, really wonderful facility. You've already heard about electron microscopy. You haven't heard that they have another specialized microscope in micro-ED that actually does microcrystal electron diffraction. So this is another example where you can look at polymers, you can look at proteins, you can look at drug substances, right? And basically, if you see on the left, yes, you have scattering. We're making diffraction patterns now, not images. So a little different than TEM for those who are not familiar with the differences. And on the bottom, what you can see is you have a tiny little crystal that's actually on your TEM grid, right? Grid, little crystal, and what you get is a diffraction pattern for it, and now all of a sudden you can actually solve the structure of that. And as we get to, again, you guys saw the examples of biology of how far it's come. Imagine when we can do that with polymers, which again are never going to have a perfect repeat crystal structure, right? Biopolymers can benefit from this tremendously, especially as it really gets off its feet. And then the last is mechanical characterization. We have an optical indenter that can give you physical characterization in 96-well format, right? We can do both viscoelastic characterization, static characterization, right? So you make the newest hydrogel that's super exciting. You want to know for sure your hydrogel is exactly this with this kind of conformity. Great. You can make 100 examples and test it all for quality control. You can also make a gradient of it and take the one that you find is the exact one that you're, or make a gradient of them, verify what they are, and then grow your cells in them and see what the effect is. So really, really great baseline for when you want to do biomechanics. On the bottom is microreology. So that's anything that's liquid, right? They say anything from water to honey, right, is basically what you can measure with microreology, and again, extract its elastic properties, right? So this is very helpful for 3D bioinks. 3D bioinks often are limited by the viscosity as they get out of printers, right? Here you could make 10 or 15 very small quantities of your test bioink, see which one actually is going to conform and be most likely to print, and then scale that up. Rheology tools usually require 3 millimeters by comparison. So with that, I would just say thank you all for listening, and I'll be available for any questions. We're downstairs, and we'll have a bunch of robots running. I always say it's like model trains. So come on down, and while the trains run, hopefully we can talk about all your exciting projects.